So channels, we talked about the transmembrane potentials, and we said that the channels are either passive or active. Generally speaking, anytime you see passive, it means we're not using energy. That's basically what it means. Active means using energy. In channels and anything, anytime you see passive and you see active, it means no energy versus using energy. So the channels can be passive, can be active. Passive means it's opened, but you can close it. The active means it's closed and you need to put some energy to open it. So this is basically what it means for the channels to be open versus closed. So the channel is either closed, but you can open it most of the time, or it's opened, you open it, or it's closed, not capable of opening. That's called inactivated. So opened, closed, or closed and you can open it. The channels in general, how do we activate the channels? How we push these channels opened? How can we open it? It depends. There are different types of channels. The first one is chemically gated, second is voltage gated, and the third is mechanically gated, meaning how to activate these channels, how to open it. Chemically is opened by chemical transmitter, basically, like acetylcholine. Voltage gated means it's activated by voltage. What's voltage? This is the transmembrane potential. Electricity, meaning electrical signal come to open it. Mechanically is distortion of the membrane. Like imagine that you're having a balloon and you squeeze it or you push it, the membrane will be distorted, right? Instead of being spherical shape, it will change. So this is going to open, like receptors, for example. Receptors are exactly like a balloon. It's spherical like this. So if you touch something, you're going to distort the shape of the receptors, and this will open the channels and will create electricity. So we have chemically gated, voltage gated, mechanically gated. Chemically, by chemical compound, neurotransmitter, specific chemical like acetylcholine. Voltage, electricity, action potential. Mechanically, distortion of the shape. So here is a chemically gated, just as an example. A chemical compound come like acetylcholine attached to the receptor and the gated channels open. Voltage gated, like this. Electrical signal come, so you see changing from minus 70 to minus 60, and this will open the gates. Mechanically, it's closed. Distortion of the membrane, you push it, apply some pressure, and it will open. These are the three types. So the transmembrane potential is what we talked about before, which is some negative ions outside, positive ions outside, negative ions and positive ions inside, if you still remember that. Now, graded potential means a local potential that come and open the gates. Okay? So what's the normal resting membrane potential? Inside compared to outside, is it negative or positive or neutral? Negative. Negative what? Negative 70. Yes. Okay? So normally, if this is the cell, you have positives inside, negatives, and then you have positives outside and negatives. If you compare this to this, if you put an electrode here, another one here, and see the reading, it's minus 70 inside compared to the outside. Okay, so if we do a curve like this, and this is like 0, 10, 20, 30, 50 until you get to minus 70. Okay, and if you started here like this is normal, right? And a graded potential come, mean electrical signal come here. Local potential. This is going to change this. The transmembrane potential is going to change. How? By opening the channels. And we talked about opening the channels before. So what's the graded potential? It's local potential that's going to open the channels. So we call this, which is negative inside compared to the outside, we call it resting membrane potential or polarized. Polarization. What do you mean by polarization? Meaning, if I have $5 here and $5 here, that's equal. If I want it to be polarized, I will have something here and something else that's different here. 5 here and 10 here. 10 here and 5 here. This is called polarization, meaning I'm being polar. I'm tending to one side versus the other. This is what I mean by polarization. Okay, so understand this first because we will talk about depolarization, removing the polarization. So the polarization is polarity, meaning one side is different than the other. If that's not the case, it's called unpolar or neutral. So normally there is a polarization. There is a difference between inside and outside. Negative inside if compared to the outside. Do you mean inside is negative and outside is positive? No. Inside contain positives and negatives. Outside contain positives and negatives. But the overall is inside comparing to the outside is negative. And this is the polarization. When an action potential come or graded potential come, the channels will open as we talked about last time. We mentioned that. We talked about how the potassium and sodium will move. What happens if you open the gates for sodium? Is it goes in or out? In. So how about potassium? When you open the gates for potassium? Out. Why? Because it's more
sodium outside.
More sodium outside and more potassium inside, right? So when you open this for sodium, it rushes in. When you open for potassium, it goes out. Is that clear? Any question about this part? So this is normal. You keep it at polarization. It's not equal. It's negative inside if compared to the outside. This is called resting membrane potential or the polarized state. It is polarized. It's not neutral. It's not equal. Polarity means you're tending to one pole versus the other, right? So when an electrical signal comes, the gates will open and you will start to lose this polarization, and this is called depolarization. Okay? Depolarization. So here it is, minus 70. Electrical signal come, the gates will open, and you will start. This is the resting membrane potential, resting state. So depolarization, you're shifting toward zero. Shifting toward zero. Meaning, this is minus 70, okay? This needs understanding. This is not something that you can just memorize as it is. It's not, okay? So you need to understand this. So electrical signal come, graded potential, and it forced the channels for sodium to open. What happened to the sodium?
It goes in.
Goes in. So what happened to the minus 70? Is it going to become minus 60 or minus 80? Which side is it moving to?
60.
Yes. Some more sodium comes in. It becomes minus 50, minus 40, minus 30, minus 20, minus 10 until it gets to?
Minus 90.
0. Until it goes to 0. It will go further, but you will go to 0 first. This is the first part. Are we following? You are removing the polarization. Sodium rushes in and sodium ion is positive, obviously. So you're putting more positive in. So it goes like minus 70, minus 60, 50, and so on until you get to the 0. Okay, so a local current or graded potential come to this cell, and it started to move from minus 70 to minus 65. How did this happen? Because of sodium. And these events are important, by the way. We have to remember each step of those. Why is it happening? Why we have the resting membrane potential that's minus 70? What happened when the local current or the action, the graded potential come? What will happen? Sodium channels open. Okay, the passive channels will open. And this is going to, sodium will rush in. Minus 70 will become minus 65 and go to minus 60 and so on. So it will go like this. Minus 70, minus 65, minus 60. So you're moving toward the zero. Until you get to the zero. And then you are removing the polarized state, right? So it goes like this. It's minus 70, minus 60, minus 50, and then it will reverse later on. So it goes like this. I will show you the picture that's very important. So repolarization means returning back to the polarized state. How to return back to the I'm going to summarize that again. But how to return back to the repolarization or the polarized state? You will open the gates for potassium at this time. So what happened when you open the gates for potassium?
Potassium comes in.
Potassium. Is potassium more inside or outside?
Inside.
Inside. So open for it. It's going to go outside. Is that going to move it toward negativity or positivity?
Negativity.
Negativity, yes. So you're returning back to the negativity, right? This is called repolarization. What's repolarization? Polarize again. So you go from polarization to depolarization and then return back to repolarization. Okay? Which is returning back to the normal state. However, we don't really return back to the normal, we even go more. So it goes like this. You depolarize like this. You go to minus 60, minus 55, and then it goes all the way to zero. It even exceeds the zero. This is depolarization. And then you start going the other way around. Okay? This is a resting membrane potential. Do you go to the minus 70 again when you repolarize? No. You go even more. Minus 90 until you go back to the normal. This part is called hyperpolarization. Not only that you're returning back to the polarity, but you're even hyperpolarizing. The example of that. Do you know the bar doors? The door when you push it and it keeps on flipping like this?
Yes.
Okay? If the door is opened like this, okay? And you open it like this and let it go. It doesn't only go back to the normal, it even exceeded, right? Is that right? So this is hyperpolarization. So depolarization, removing the polarity, repolarization, polarize again, but it doesn't take it exactly to the original point. It even exceeded until and then go back to normal. This is like the bar door, open it, let it go, it goes back and even exceeded and start to flip like this and stop, right? So this is the stop point right here. So it is going to go back to the minus 70 again, but after exceeding that. So what happened when the graded potential came or a signal came? At the dendrites and the cell bodies, do you remember the parts of the neuron? At the dendrites and the cell body, which is the first part, special functions will start, like exocytosis. If this is a motor end plate, acetylcholine will be released. What's the motor end plate? Yes. So between nerve and nerve, you just call it synapse. The motor end plate is at the muscle. Okay? So if that at the muscle, acetylcholine will be released. So action potential is going to move move from spot to spot to spot, propagating until it goes to the end of the neuron. Okay, so the initial stimulus. Graded potential came. Depolarization. Graded depolarization occurred at the axon hillock. Do you remember the axon hillock? Do you still remember the parts of the neuron? Okay, so when a graded potential comes, here is what's going to happen. Graded potential come here, right? It's minus 70. It's going to go to like minus 65, minus 60, minus 55. If it goes to minus 65, that's enough to continue the process. If the signal is not strong enough, let's say take it from 70 to 65, it will go back to the polarization. It's not going to continue the whole cycle. So the point I'm trying to make here is, if your initial stimulus is strong enough to cause 10 to 15 millivolt change, that will continue the whole process. If it's less than that, it will not continue the whole process. And this is called the threshold. Are we following? This is like the hardest part of this chapter. So if you're not following, that's fine. Just let me know and I will explain it again. Okay? Okay. The cell normally is in polarized state. Polarization. This is normal. It's polarized. It's not neutral. It's negative inside compared to the outside. Okay? All right? Okay. We have channels. These channels are either open, normally open, we call it passive, or it is closed and you can open it, and this is what we call active. Are we following so far? I'll take it step by step. Here is your cell. Measure the cell and tell me. Okay, I measured it. I found that you have positives and negatives inside. You have positives and negatives outside. Measure it. Put the electrode. I put it and I found that it's negative inside of compared to the outside. How negative? Minus 70. This is normal. Nothing is happening. Are we following so far? Okay. So now a signal come to the cell. This signal is going to open the passive channels for sodium. What happened to the sodium? Goes in. What happened to the minus 70?
Goes down.
To I would call it going up, but whatever the way you get it, because less negatives means positive, right? Minus 70 become minus 60. If you're moving towards zero, actually you're increasing. But anyway, minus 70 will become minus 65, right? Minus 60, minus 55. What's happening? Why it's doing like this? Because the passive channels for sodium are opening. Okay? So this change from 70, sodium getting in, minus 65, getting in, minus 60, getting in, minus 55. If you can take it all the way, if you can keep it open until you get to minus 60 or minus 55, the next channels which are active will open. Are we following? Are we following so far? So stimulus come, passive channels open, sodium rushes in. To what extent? To the extent of minus 60 or minus 55. Can you take it all the way to minus 60 or minus 55? If yes, the active channels will open. And if the active channels will open, it will continue. So this is the maximum that the passive channels can open. It can give you a little bit of change. It open and allow some sodium to get in. So depending on the strength of the stimulus, the open channel can go from minus 70, minus 65 maybe. That's nothing. minus 60, or minus 55, that's strong enough to open the active gated sodium channels. So what happens when you open the active gate? It will wide open and all sodium rushes in. Are we following? So it is moving from minus 70, stimulus come, minus 65, minus 60 or 55, okay, that's good enough. The active channels will open and it will shoot all the way to minus 50, minus 40, 30, 0, and even more, right? So that's what's happening for depolarization. And what do you call this? 10 or 15 change, that was enough. So this part is by passive channels. If you exceed that, the active channels will open. What did you call this point? Threshold stimulus. Threshold. You exceeded the threshold. What's a threshold? 10, a change of 10 or 15, or you take it to minus 55 or minus 60. This is the threshold. You exceed the threshold, the active channels will open. You don't exceed the threshold, it will not open. Are we following so far? Okay. This is for depolarization. So, all or none principle, and this is important to know. What's the all or none principle? You either have a stimulus that exceeds the threshold or not. If you exceed the threshold, active channels will open, you will have this cycle. If you don't achieve, or if you don't exceed the threshold, it will not open. So we don't have something in the middle. It's either strong enough to complete the cycle or nothing will happen. All or none rule. Okay? So you either produce action potential or not, which is this. So let me ask you this. A stimulus came to the cell is minus 70, a stimulus come and it make a change of 8 millivolt. Is that going to produce action potential or not? No, that's not strong enough. Did you exceed the threshold? No. How about 15? Yes. Why? What happened at this point?
More than 10.
It's more than 10, active channels open. So what was the first part? The 10 or 15, what is this? What caused it? What changed from minus 70 to minus 60 or 55?
The potassium.
Not the potassium yet.
Sodium going in.
Which channel? The passive or the active? The passive. Are we following so far? Okay. So here is minus 70, and you go to depolarization. You got to the threshold. You activated sodium channels. That will cause rapid depolarization. If you compare this part versus this is called slow depolarization, and this is called rapid depolarization. So the first part before you get to the threshold is called slow depolarization. Isn't it both of them depolarization? Aren't you removing the polarity anyway? Yes. But the depolarization that's caused by the passive channels is very slow. So sodium leaks a little bit. Okay? If you open the active, it will rush in. So this part is slow and this part is fast. What do you call all this part? Depolarization. Are we following? So until this point, this is all called depolarization. Okay? So you cause this minus 60 or minus 55. Remember both numbers, it's either one of those is fine. Minus 60, minus 55, or the other way around, which is 10 to 15 change. This is good enough to open the active channels and start the phase of rapid depolarization. Okay? Okay, so if you take it like this, 50 minus 40, and this is minus 30, minus 20, minus 10, 0, 10, 20, 30. So look what happened here. You don't only go to the 0. You even exceed it. You go to positive 30. Are we following? Okay? So you take it all the way to positive 30. What's the positive 30? Why do you want to take it from minus 70, minus 60, minus 50, not only to the 0? You go even positive 10, 20, 30. What's the magic number of 30? This is the threshold for the potassium channels. We didn't mention any potassium channel so far, right? We're talking about sodium. So now, when you get to the positive 30, this is the potential that's needed for the potassium channels to open. What happens when the potassium channels open? Rushes in or out?
Out.
Out. So aren't you going back to the polarized state? Right? When you move the positive out, aren't you making the inside negative again? Right? When I take the positive from you, aren't you moving to negative? Yes. So positive will rush in, which is potassium, and this is called, what do you want to call it? Repolarization. You're going back to the polarized state. Is that true?
Yeah.
Right. Okay. So what is this part?
Repolarization.
Repolarization. So you repolarize, meaning positive 30, positive 20, 10, 0, minus 10, minus 20, minus 30, minus 50, minus 70. Is that it? No, you even exceeded more. So what do you call this part? Hyperpolarization. It's not only repolarization, you don't only return back to the normal, no, you even exceed it. It's called hyperpolarization. So what's the cause of this part, channel-wise?
The threshold.
This is the threshold, but channels.
Well, the potassium. Sodium.
Which ones?
The passive.
The passive. How about this?
Passive. Still passive.
Active. This is passive. This is active. This is what the threshold is all about. When you reach the threshold, at this point, the active will open. Okay? And what's the cause for this? Which channels? Potassium. Are we following or not? And what do you call this part? What do you call this part? I'm talking about the depolarization now. Slow or rapid? Slow. And this?
Rapid.
Rapid what?
Depolarization.
Depolarization. And what is this? And this? Are we following? Okay. So this is very important, by the way. And you will see, you can actually see this curve. Clear enough? You can see this curve. And that's why I'm spending time to explain it. Not just because it can show up in the exam, but because it's very important to understand. We have to understand. So you're returning back to normal when sodium channels open and you go back. At this point, what will happen? Potassium channels will close so that you return back to normal. Okay, so did you start at minus 70?
Yeah.
Did you end at minus 70? Yes. So you return back to the normal state? This is from the electrical point of view. But think about it. Did you go back to the normal state regarding potassium or sodium? Or it is upside down now? Think about it. What's normal? Sodium more outside or inside? Sodium. Potassium? Both of them inside?
No, sodium outside, potassium.
Sodium is more outside. This is normal. Potassium is more inside. This is normal. What did you do now at the end of the cycle? You reversed. You reversed the whole thing. Didn't you open the channels for sodium? Didn't sodium move in? Right? So it's not more outside anymore? Didn't you open the channels for sodium, for potassium? Potassium moved out. So now potassium is more out and the sodium is more in. Is that normal? No, it's the opposite, right? So yes, you return back to the normal from the electrical point of view, from the voltage point of view. I started with minus 70, I go like minus 60, minus 50, 0, positive 30, and then I go back to normal minus 70. Yes, you did. But how about the ions? Did you turn back to normal? No, you did not. You actually, yes, you achieved it in another way, but now you have it upside down. How to solve this problem now? You have an enzyme. Now you're back from the voltage point of view, from the electrical point of view, not from the ionic point of view. So now I wanted to reverse it back to normal, to be completely normal. How to do that? There is an enzyme that's called sodium potassium ATPase that will take sodium out and potassium in. So now you return back to normal. So when I ask you what's the normal, you don't only say the normal is minus 70. This is from the voltage point. Yes, it is. Minus 70. This is normal. But how about the ions? Normally, the ions, sodium is more outside, potassium is more inside. When you do this cycle, you are reversing it. Did you go back to normal? Yes, from the voltage point only. How about the ions? No, it's upside down. You reversed. Can you return it back to normal after you do the cycle? Yes, by the help of an enzyme that's called sodium potassium ATPase. What sodium potassium ATPase will do? ATPase, meaning it's going to break down ATP to get energy out of it, and it's going to reverse. Okay? It's going to take sodium out and potassium in. Sodium out, potassium in. Sodium out, potassium in a ratio 2 to 3. We will talk about this. Okay? Now, if you see a curve like this, can you solve it if I put like A here? Do you know what is this?
Yeah, slow depolarization.
Slow depolarization caused by
Passive channels.
passive channels. And what is this?
Rapid polarization.
Caused by?
Depolarization caused by active channels.
Yes. How about this point? What happened here for the channels? This is the threshold. What happened for the channels at this point?
Open.
What opened?
The sodium.
Which one? Active channels open at this point. What happened at this point? Potassium channels open. And what's happening at this part?
Polarization.
Repolarization. What caused it? Potassium moving in or out? Out. And until you get to the resting state. What is this?
Hyperpolarization.
Hyperpolarization. Are we following? Okay, so this is very important. I think I'm clear enough. So expect to see something like that. Okay, so now, how about this? You started the whole cycle and you get back to normal, okay? After you turn back to normal, everything is back. It's minus 90. Sodium is more outside, potassium is more inside. Isn't that completely normal? You return at this point. You return back to minus 70, right? So minus 70. And you use the enzyme sodium potassium ATPase that took the sodium back outside and potassium back inside. That's normal, right? How about another signal come at this point? Okay, so let's go this way. Another signal came. It's moving like this. Is it going to produce another action potential? Or not? What's your answer? It depends. Is it strong enough to reach the threshold or not, right? If it is, if you go to minus 60 or minus 55, then yes, the cycle will go again. If not, it's not going to happen again. That's number one, all right? How about this? What if another signal, instead of coming after the whole thing is done, what if it come here? or here, or here. Okay, another signal came prematurely before you finish. It depends. How early did it came? So there is a period that's called absolute refractory period. And understand what does it mean? Absolute refraction. It's completely refractory, completely resistant. It's not going to respond. So what's absolute refractory period? This is when the sodium channels opened or inactivated and no action potential will occur. It's not going to respond to you. Absolute refraction, absolute refusal. Nothing will happen, no matter how strong the signal is. This is called the absolute refractory period. Relative refractory period means you are very close to normal. So if a stimulus come at this time, which is you're going back close to normal, now it depends on the strength of the signal. If it is large enough, it can produce. If not, it will not produce, and this is called relative refractory. So what do you mean by relative refractory? Meaning relatively refractory. So it refuses until you give it enough signal. But the absolute, it doesn't matter. Okay? So absolute refractory period versus relative refractory period. Absolute refractory period, the period of absolute refraction. No matter how strong the stimulus is, nothing will change. The channels will not respond, irrespective of how strong it is. Why? Because it's already opened or inactivated. So you can do whatever you want. It's not going to respond. Absolute refraction. How about the relative refractory period? It's a period of relative refraction, relative refusal. So I'm refusing to respond except if you give me strong enough stimulus or large enough stimulus. Are we following? This is the refractory period. And why do we have the refractory period? To avoid repetitive stimulation that's enough to make bad effects. Meaning, you want your muscle to contract and relax, right? Contract, relax, contract. You can't go like contract and another signal come, contract, contract, contract. You can't do that. That's called tetany, right? You can't just contract, contract. You have to give the muscle a little bit. Not necessarily all the way to complete relaxation, but just give it a little bit time to relax. When the muscles start to relax a little bit, for example, if we're talking about the muscles, it will have enough time to get some energy and some calcium. You can't like stimulate repetitively, and the muscle is responding to you all the time. That will go like this. This is called tetany, okay? Which is, you don't give it any time to relax. So it will go like this until actually it will lead to death. And this is what tetanus is all about. A lot of stimulation, and it goes like not relaxing at all. Contraction, 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 until the muscle fall down. Okay? That's why we have the absolute refractory period, which is a little bit of time that you cannot stimulate the channels. So the sodium potassium ATPase enzyme does it in a ratio of 2 potassium to 3 sodium. Okay? It goes like this. I am the sodium potassium ATPase enzyme. Okay? I'm the enzyme. I'm going to move sodium back outside and potassium back in, right? Every move, I'll do like this. Okay? 2 out, 3 in. Okay? Or the other way around, I'm sorry. 3 out and 2 in. So 2 potassium and 3 potassium out. Why did you do that? This is the nature of the enzyme. Okay? So the enzyme is like this. Just imagine this is the enzyme. Oops. This is the enzyme. You have this. This is the configuration of the enzyme. So it can get 3 and 2. 3 sodium and 2 potassium. It's going to reverse. Okay? Flip it, 3 and 2. Flip it again, 3 and 2. Flip it. This is just a configuration of the enzyme. So always take 2 potassium in, 3 out. 2 in, 3 out. 2 in, 3 out, until you return everything back to normal. All right? What if you don't have ATP? You can't do that. So it's not going to respond anymore. And that's when we run out of energy. Okay? Okay. So this curve is extremely important. This is the absolute refractory period. Start from this point to this point. You see the purple? Okay? So any signal that come in this period here, this is absolute refraction, nothing will happen. It's not going to respond. How about this blue? Look at this part, which is number 4. This is relative refractory, meaning if a signal come here, or here, or here, or here, if it's strong enough, it's going to respond. Okay? So this is extremely important. Okay, this is in the core of chapter 12. So of course expect to see questions and expect to see maybe something like this. And I can ask you at number 1 or 2 or 3 or 4, or ask you what is the whole thing here or the whole thing here. What's number 1?
Slow depolarization.
Slow depolarization, yes. What's this point here? Resting. What's number 1? Slow what?
Depolarization.
And what's number 2? And what's 3?
Repolarization.
What's 3 exactly? This is repolarization. What 3?
Potassium channels open.
Potassium channels opens. And what is this? Purple?
Repolarization.
Repolarization. And what is this?
Hyperpolarization.
Under the line? Number 4? Hyperpolarization. And what is it from here to here?
Absolute refractory period.
And this? Relative. Are we following? Okay, so this is important. And then after you do that, so you started an action potential, and the action potential is going to propagate, and the neurons are one way. Do we go from axon to body to dendritis, or from dendritis to body to axon? What do you think? We talked about the neurons.
Dendritis to body to.
Dendritis to body to axon. Can you go the other way around? No. You can't go to the body and then the dendritis, you cannot reverse it. It's one way. Okay? Yeah, you can move in a little bit, but you can't take it all the way to the opposite way. Okay, now we mentioned before that we have, if this is the neuron, dendritis, this is the body, and this is the axon. What was this? Collaterals, yes. And this? Telodendron, what is this? Yes. Okay, so the axon itself can be myelinated or unmyelinated. Which one is faster? If you're moving the electrical signal, unmyelinated or unmyelinated, which one is faster? It is actually myelinated. I know you think it's not myelinated, so there is nothing in the way, so it moves faster, but that's not the case. The case is if you're jumping or walking. This, you're walking slow. And this, you're jumping from this to this to this to this to this. Yeah, if you have the myelin sheath, you'll go like this. This is electrical signal. Jump, 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 jump. You get to this point in 5 milliseconds, let's say. Versus if you're here, you're walking 10 milliseconds. Okay, this is how it goes. Are you walking or jumping? Right? So which one is faster? Myelinated. And this is called saltatory. Saltatory means jumping. Okay? So saltatory propagation and myelinated, and this is faster. Versus continuous propagation, unmyelinated, and this is slower. Clear? Okay. So continuous versus saltatory. Okay. What I'm summarizing now, this is the most important thing, and you can go back to the recording and see what we're talking about. This is what you have to know. This is the core, okay? So Otherwise, if you wanted to read more, that's fine. But what I'm telling you is the core of the chapter, and this is what you have to know. Okay? So now if you're comparing the graded potential versus the action potential, the graded potential is the first part, okay, which is depolarization or hyperpolarization. The action potential is depolarization. Okay, second part. How about the axon diameter? So in the first part, we talked about, is it myelinated or unmyelinated? Which one is faster? Myelinated. What do you call it? Saltatory. Saltatory propagation. And if it is unmyelinated, slower, and you call it continuous propagation. Are we following so far? Okay, now the axon diameter. Which one is faster? The large diameter or the small diameter? Large diameter. Why? Because if I put you inside a tube that's large like this, can you run? Yes. If I put you in a tube like this, can you run? Possibly not. You have to squeeze yourself in and move slowly, right? So the larger the diameter, the faster the movement. So we have two components of the speed so far. The axon diameter and myelinated versus unmyelinated. So according to that, we have, according to those two factors that I just mentioned, you can have three types of axons. Type 1, type A, B, and C, depending on the diameter and myelination, and this will give us the speed. Type A, what do you think? This is the best. So what do you think? Is it myelinated or unmyelinated? Myelinated. Is it large diameter or small diameter? Large diameter, and this is the fastest speed. Okay? And the example here is very important. So if I'm doing like, I'm building a house, right? I have different types of wires. This is large wires, high quality wires, and this is medium, and this is low quality wires. I'm going to use obviously the high quality large wires in the large things, the more important things like the fridge, for example, or washer or something. I'm going to use the medium one for like the medium stuff, light bulbs or something, and the small one I'm going to use it for trivial things, right? That's wise, right? So how are you going to use the type A? I'm going to use type A for the most important part and the fastest part, the part that needs really fast reaction, like position, balance, touch, motor impulses, and you need to remember this. Meaning, I cannot afford, and you cannot afford to lose your balance or position for a millisecond. You will fall down. Can you afford that? If you lose your balance for less than a second, you'll fall down, right? So this needs immediate response. So I'm going to use the best type that I have, which is type A. Balance, position, touch. Type B is myelinated still, but it is medium diameter. So something like sensory information and peripheral effector, which is the afferent and efferent. Sensory information is sending, it's still fast, but not as fast. Okay? So this is type B. Versus type C, which is unmyelinated. This is the only one that's unmyelinated. Okay? And this is the slowest of all. And something like involuntary muscles or gland control, for example. Can you have an emergency sweating, for example? I have to sweat as soon as possible. Do you have that? Of course not. This is something that's, you gave it the least important ones. The slowest one. It's not urgent. What's urgent about that? Any gland, whatever it is. Okay? Sweat glands. Ceruminous glands. Do you have emergency producing the wax, for example? No, it can take as much as it needs, right? Involuntary muscles. Do you have an emergency that you have to contract your stomach, for example, to move the food down? Does it make a difference, a second or 5 seconds or even 5 minutes? Doesn't make a difference, but balance and position, no. Not even for a second, right? So what I want you to know here is type A, type B, type C. A and B is myelinated, C is unmyelinated. A is large, B is medium, C is small, fast, intermediate, slow, and examples. Where is it located? If you follow what I'm talking about, and when I focus on something, you get it write it down, do whatever you want. Trust me, you will do very good in your exam, okay? Because I focus more on the core of the chapter. It's not all the same. Do not study everything the same way. Spend your time in everything. That's not right. Time management, how to manage your time. This is, and I'm recording it, so you can return back to it. Whatever I'm focusing in is the core of the chapter. You have to know it. Like a comparison, I'm giving you a comparison. You cannot miss it in your exam, right? But other things, just read it. Do you know how to do the time management? Okay, this is how. If you forgot this, go back to the slides. Yes?
Exam number 3, is it only chapter 12 and 13?
Yes. I'm expecting you to do good. And again, this is how the game work, okay? Follow what I'm focusing on more. This is the core of the chapter. And because it's the core of the chapter, expect to see more questions, right? So follow this. You forgot, go back to the slides.
So is our exam 3 going to still be the same date as on our syllabus?
I'm not sure. It might, if we're behind, I might postpone it for one day or something, depending on the situation. Okay. Are we supposed to finish at what?
9:30.
9:30?
Yeah.
Okay, good. Okay, so now you're propagating. We talked about propagation already, and you follow, right? Type A, type B, type C. Now you are propagating irrespective if it's A or B or C, and you reach to the end of the neuron, the end of the axon. Now, if the next part is called the neuron, if this is the end of the neuron and this is the beginning of another neuron, the part in between is called the synapse, and this is called presynaptic, okay? And this is called postsynapse, okay? So you're crossing the synapse now. There are different types of synapse. Electrical synapse, like this. These are the dendritus, body, axon. Dendritus, body, axon. It goes like this, right? If the dendritus touch the teledendria, this is electrical synapse. If it doesn't touch, then you have a gap, then you have to use a chemical transmitter. You call it chemical synapse. Is that clear? Okay, so the electrical signal is direct physical contact. Here is the end, and this is the beginning. It touches. Electrical synapse. If it doesn't touch, you have a synapse in between, and you have to use a chemical transmitter to transmit this or to cross the synapse for you, and this is when we call it the chemical synapse. Electrical synapse is also called gap junction or connexons, and it's important to know where it is. Because this is the exception. That's not normal. I shouldn't say normal because it's all normal. I mean, this is the vast majority is chemical. So the exception is electrical. Where do we have electrical? Brain, eye, ciliary ganglia. And you have to remember that. These are the 3 locations where you find the electrical synapse. Do you mean that everything else is chemical? Yes. Anything that's not those 3 is chemical. You have to have a gap, you have to have a synapse, and you have to release a chemical transmitter that cross the synapse, go to the postsynapse, open the gates, start an electrical potential, and so on. It moves from 1 to 1 to 1. So this is normal, chemical, or this is the vast majority, I should say. But the electrical signal, which is like this, direct physical contact, this is only in the brain, eye, and ciliary ganglia. The chemical synapse is everywhere else. And this is where we use a chemical. Okay, so when you reach to the end of the presynaptic neuron, and you're going to release a chemical transmitter, and this chemical transmitter is going to travel across the synapse for you and go to the postsynaptic neuron. What we're imagining right now is the neurotransmitter go to the end, cross the synapse, go to the postsynapse, produce an action potential, and it's not true. Most likely, yes, but sometimes it inhibits the postsynapse instead. So this is called excitatory versus inhibitory.
explain the post-synapses and pre-synapse?
Yes. What is this? Dendrites. What is this?
The body,
What is this?
the axonal, right? Isn't that what it is?
Yeah, but what's the next?
The axon.
The initial segment?
Initial segment of the axon. And then the axon itself. And what is this?
Collateral.
Teledendria, synaptic terminus,
And what is this?
synaptic.
The synapse. Or the synaptic cleft, if that's what you're talking about. And what is this again? And you're moving one way, right? You can't move this way. So if you're moving this way, this neurons is before the synapse. And this neuron is after the synapse, right? So this would be called presynaptic neuron, and this is called the postsynaptic neuron. Did you get this? Okay.
So would you only know which one's the pre and the post if you were comparing the two?
The way to know is, are we moving to the teledendrious synaptic vesicles, to the dendritis? So you have to either see it or it's being described to you, okay? Because it only goes one way. So if I'm telling you I'm at the synaptic vesicle of what? Presynapse. If I'm talking about the dendritis, it has to be the postsynapse, right? Depending on the location. So now the chemical transmitter is going to be released. What is this again? Synaptic what?
Terminal.
Synaptic terminal, synaptic knobs. It's the same,
Yeah.
okay? And what do we have inside this?
Neurons.
Neurotransmitter. So the electrical signal will move down the axon, go to the synaptic knobs or synaptic terminals. This neurotransmitter will be released, cross the gap, cross the synapse, go to the dendritis, and actually it can go to the body. It's crossing the gap anyway, going to either dendritis or to the body, whatever, okay? And this neurotransmitter, when it goes to the body or the dendritis, it can either produce another action potential here, which is normal, and then the action potential will move the same way. Neurotransmitter, cross, another action potential, propagating. But sometimes it's called the inhibitory. So it go to the body, let's say, and instead of producing an action potential, it actually lock it. No, you're not going to respond. I'm here to tell you, don't open. So this is another type of neurotransmitter that's called inhibitory. What exactly is happening here? Here's what's happening. Neurotransmitter is released. What's most likely the neurotransmitter is? Acetylcholine, right? 90% of the time it's acetylcholine. So you have to remember this. Acetylcholine is crossing the gap, right? How is it released in the first place? Here's the electrical signal moving down this way, right? Until it reaches the synaptic vesicles, the synaptic knobs. And the synaptic knobs, I'm going to magnify one of these, okay? So let's say this is the teledendria, and this is the synaptic knob. Just magnifying this, okay? Right? So we have synaptic vesicles, like pouches, that contain the synaptic neurotransmitter. And I said it's going to be released, right? It's inside a pouch. What released it? How is it released? Can somebody tell me? Why is it released? Is it just released by itself? There has to be something that burst or opened the vesicles and released the acetylcholine, which is a neurotransmitter. What happened here?
Don't they fill up?
It's filled and staying like this.
When they can't fit no more?
It can't fill no more. So what opened it? Why the neurotransmitter is released out of it? When the electrical signal come to the end at the synaptic terminal here, it will open the gates for calcium to get in, and the calcium is what's going to open the vesicles. Okay? Are we following? And the synaptic vesicles will open, release, going to the postsynapse. It's going to react with a receptor on the postsynapse. Are we following? When you react with the receptor, sodium channels will open. Sodium gets in. What do you call this? Sodium gets in. Depolarization. So aren't you producing depolarization here? Right? If it's strong enough, what happened? You're producing?
Rapid depolarization?
which is called, the whole thing is called depolarization, repolarization. The whole thing is called action potential. So you produce action potential here, right? That's going to propagate, go to the end. Calcium comes in, right? Opening the vesicles, release, go to the postsynapse, react with receptors. Sodium channels open, sodium gets in, depolarization, action potential, and moving on. Okay? Are we following so far? So what are you producing here? Depolarization, right? How about this? Another neurotransmitter that come react with another receptor, and instead of opening the gates for sodium, it actually opened the gate for potassium. Is that going to cause depolarization?
No.
No, it's going to do the opposite, which is hyper. Yes, more repolarizations, hyper. So this is the inhibitory neurotransmitter. Did you understand this? This is the inhibitory neurotransmitter. So what's the action of excitatory versus inhibitory? Excitatory neurotransmitter is going to open the gates for excitatory, open the gates for excitatory, sodium, inhibitory, potassium. Okay? Okay. What's the fate of acetylcholine? Here is acetylcholine released from the vesicles, synaptic vesicles. What's released it? What opened the vesicles?
The calcium.
Calcium. So now the neurotransmitter is released, which is acetylcholine, crossing the gap, reacting with the receptors, opening the gate for sodium, or sometimes potassium. If it opened the gates for sodium, what do you call it?
Depolarization?
And what do you call the neurotransmitter that caused this? Excitatory and inhibitory? Excitatory. And if it opened the gates for potassium, you call it inhibitory. So okay, so now the neurotransmitter crossed the gap, reacted with the postmembrane, everything is fine. What's the fate of acetylcholine at this point? You have to break it down. Why? What if you didn't break it down? What will happen? It keep on stimulating. You don't want to do that. All what you want to do is acetylcholine cross the gap, interact with the receptor, causing depolarization, and go away. I don't need you anymore. But if you leave it, if you didn't break it down, it will go like this. Stimulate the receptors, depolarization. Stimulate the receptors, depolarization. Stimulate the receptors. You don't want to do that. You just want it to go one time, stimulate the receptors, cause depolarization. Thank you. Break it down. How do you break it down? What's the enzyme that break it down? Acetylcholine. It have the same name?
Acetylcholine esterase?
Yes, acetylcholine esterase. Okay? So acetylcholine, you released, goes to the postsynapse, react to the receptor, open the gates for sodium, cause action potential. That's it. Thank you. Break it down. Break it down by what? Acetylcholine esterase. And acetylcholine esterase is going to break it down into very simple. Acetyl and choline, right? Acetylcholine, break it down into acetylcholine. Acetyl is cheap. We don't need it. You know when you get a can of soda or a bottle, okay? The cap, usually we throw it away, and you keep the actual bottle because of the deposit, 10 cents or whatever, right? So this part you can recycle it. Do you recycle the whole thing? Most likely, or not necessarily, right? You can throw away the cap, and can you still recycle the bottle itself?
Yeah.
Yes. So the bottle itself is choline. The cap is acetyl. So acetyl is cheap. You don't need it. Just get rid of it. But recycle which part? Choline. This is the important part. This is the expensive part. This is the part that you can actually reuse, and it's actually expensive. The body spends some time and energy to make it, so don't throw it away. What are you going to do when you break it down? Get rid of acetyl, we don't need it, but return the choline back. Recycle it. return it back to the vesicle. And then what happened here? The synaptic naps will say, okay, so the acetylcholine that I produced, cross the gap, you did the job, thank you, break it down, get rid of acetyl, choline return back. I have a lot of acetyl in there. I'm going to put it back and make it again. Recycling the acetylcholine. Did you get this? Okay. Okay, now we said that most likely the neurotransmitter is acetylcholine, right? Do we have other neurotransmitters? Yes, but most likely it is acetylcholine. So where is it located? Actually, when the synapse is using acetylcholine as a neurotransmitter, you call it cholinergic synapse. So what's a cholinergic synapse? A synapse that uses acetylcholine, okay? Where is it located? Neuromuscular junctions, synapse in the CNS, any neuron to neuron in the peripheral nervous system, muscular and neuroglandular junction on the autonomic nervous system, parasympathetic division, not the whole autonomic. What's autonomic? What are the two divisions of the autonomic? Sympathetic and parasympathetic. Parasympathetic is using acetylcholine. How about the sympathetic? No. It's using adrenaline or epinephrine, norepinephrine, right? So this is actually most of the synapses. But you still have to know this. Okay? Which synapse release acetylcholine? Most of the synapses. You still need to know this, but the important part here is not the sympathetic autonomic. What do we use in the sympathetic autonomic? Adrenaline or noradrenaline. What's the other name for adrenaline, noradrenaline? Epinephrine or noradrenaline. What's the other name for noradrenaline? Norepinephrine. Okay? Okay, so we talked about this already. And this is explaining what I was just talking about until the action potential is produced in the postsynapse. The synaptic delay, the time it takes from the arrival of the action potential until you release the neurotransmitters and do the action. So this part of crossing the synapse, it is very tiny, but we still have to know it's by milliseconds, not even a second. But it is about half millisecond. Remember this. The delay is the time that takes. This time is very, very small. It's 0.5, not even 1 millisecond, but it's a delay anyway. And this delay, well, what make it 0.2 and what make it 0.3, 0.4, 0.5? This will depend on how many synapses do you have in the way. So you add up. Okay. I mean, let's say this is a neuron, okay? And then delay, neuron, delay, neuron, delay. So the more synapses you have, the more delay you have. Is that true? So if this is 0.5 milliseconds, and I have 10 synapses, that will take 5 milliseconds until you go to the end, right? So the more synapses, the more delay that we have. That's why the reflexes usually involve one synapse only. Are we following? If I wanted to move from point A at this wall to point B at the other wall, and I have different neurons in between, okay? If you wanted to move it fast, make 2 neurons only, from point A to point B. So it goes to the neuron, one synapse, next neuron. That will take 0.2 to 0.5, right? Versus, what if I put 10 neurons from point A to point B? It will be 10 delays, right? So it will take more time. So in case of reflex, do you want a lot of delay? Of course not. Why? Because the reflex is the basic function of the nervous system, meaning if you touch something that's hot, every millisecond means something, right? You wait, millisecond, you're burning your epithelium, right? Give it just a couple more milliseconds, it will burn the whole thing. Electrical signal, for example. What if you touch electricity? Can you afford the delay? Can you wait even for a millisecond? You can't. You have to move your hand right away. And that is the reason why the reflexes will have less. It only have one. Just one goes in, one goes out. You can't have a lot in between because of the delay is something that you cannot afford at this time. Synaptic fatigue is if you are using the synapse many times, you need to give it some time to get some more energy and replenish the acetylcholine, meaning you're going to the end, release acetylcholine, break it down, take back choline, get acetyl and put it on it, right? Now it's ready to be used again. Another signal come, release it, do the work, break it down, return back choline, put another acetyl. Doesn't that take some time? So if you send signal after signal after signal after signal, you don't have the time for the acetylcholine to recycle, to replenish. So if you do that, they will come to a fatigue, synaptic fatigue. Okay, let's stop here and we'll do the rest next time.